So today we will talk about the COVID-19 analysis in Galaxy, and specifically the importance of an existing open infrastructure to respond to this pandemic. In a nutshell, this is what we will be discussing of the different aspects of this infrastructure. On the left, we have the data that we need, that needs to be available, for which at the European side, the COVID-19 data portal is instrumental. And we've integrated that into Galaxy to do analysis and to make this data available for other projects according to standards that we have been developing in projects such as EOS-Live and ELIXIR in collaboration with the Galaxy community. So today we have 5 speakers. Nadim Rahman and Guy Cochran will introduce the COVID-19 data platform. Björn will talk about the COVID-19 analysis in Galaxy, and then Roloni will specifically demonstrate how they did this in Australia in collaboration with the rest of the world. And I will wrap up by placing this in an integrated ecosystem that we have rolled out globally. So with that, I give the floor to Guy Cochran on the COVID-19 data platform.
Great, thanks very much, Frederick. So let's move straight on to the next slide, please. So we launched the European COVID-19 data platform back in April last year, in the early days of the pandemic. And it's an effort that is a partnership between many, my institution, the EMBL, European Bioinformatics Institute, ELIXIR, European Open Science Cloud, and it's generously funded by the European Union under a number of different projects that you see on the right. And also we partner in this with many clinical, public health and academic partners to deliver all the different parts. So it's a very big project. It has parts that are centralized, that operate at the national level, connections into the European level research infrastructures, and global connections in many different ways. And the function is really to provide the data foundation with a focus on molecular biology data around which COVID research can take place and we can fight the disease. So if you look at the top left here, these are the things, some of the components that are organized centrally. We have the COVID-19 data portal and the SARS-CoV-2 data hub system. And I'm going to focus in particular on these. These are the components that are relevant for the work that will be described by others later on in this webinar, but also in the future webinars to come as well. These are the feeder layer for some of these activities. So if we go to the next slide, please. So this is the component of the platform that is the COVID-19 data portal. And what we do here is we bring together many different data types of relevance to COVID-19, and we organize them and make them available through a single interface. This isn't a big data, well, it's a database, but it's not an enormous database of everything. Rather, it is a view on many different pre-existing and some new databases. And we very much leverage here the ELIXIR core data resources, or the ELIXIR deposition databases as the source data. And really, without a history across these databases of working openly with open data and providing useful practical interfaces, programmatic interfaces into those data, it really wouldn't have been possible to launch so quickly. So the launch in April was very fast, but it really sits on the fact that we have this great history of fair and open data collection and provision that we could leverage. And you see the bit of the timeline there, it's not going quite up to the present day. We've added some data resources since then, but very early on, we were able to connect in data of different types. Of course, there were many data sets that were directly relevant to COVID-19 in advance of the outbreak. But of course, there's great effort to capture new data that relates specifically to the outbreak. And this brings both of those components together. And I think actually the fact that we start with this means we can drive openness in data as well. So open data, really critically important in what we're doing here. If you look at the bottom, you see the different data types we're collecting. Many of these are sequence or sequence derived, but we also have proteomics data, we have protein structural data, imaging data, data about protein complexes or macromolecular complexes, compounds, and so on. And then, of course, the literature. So many different data types that are important available from a single portal. And if you look at some of the numbers, this slide I prepared only a couple of weeks ago, but already some of these numbers have changed dramatically, particularly on the sequence side. So if we go on to the next slide, please. Nadine will say a little bit more about this. So the portal provides different interfaces. There's the website, of course, that allows navigation. There is a top-level discovery API, but each of the underlying data resources has at least one more specific, more granular API for access. And there are different ways of syncing across, for example, FTP endpoints as well. So on to the next slide, please. So the portal is drawing in data from lots of different underlying databases. And I'm going to take the example of the European Nucleotide Archive as one of those databases we connected at the beginning. I take this example because it's used for sequence data of relevance to the pandemic. Importantly, as we will discuss later, it's used for viral sequence data. It's also used for data that are freely consented for open distribution. So data that are not sensitive from humans. But also data from animal studies and so on. So it's both virus and host. And I guess many people, I hope, will be familiar with the ENA already. I guess the thing to point out here is that this is not just a single database that sits in Europe. It is connected to a global exchange network where it works with partners in the US and in Asia on a regular basis to exchange data. So putting data in here immediately makes the data available to the rest of the world, but also it benefits from capturing data through these partners from elsewhere in the world. So if we move to the next slide, please. So putting data into ENA, of course, is really only the beginning of a journey that a data set takes. Here on the right, we're looking at the different ELIXIR core data resources. And if you see at about 1 o'clock there, you see ENA. As soon as a data set arrives in ENA, immediately, depending on the data type, there is a data flow into many of the different ELIXIR core data resources. And these things are autonomous. These happen. We've set these things up a long time in advance. And so, for example, assembly data would flow to Ensemble. We would have protein data flowing to Uniprot, for example. So immediately the data go in ENA, there is a flow into this ecosystem of connected databases. And it's really the openness that is the lubricant to this. So if we move to the next slide, please. This takes it one step further. So this is a recent review of the database issue of nucleic acids research and the collection of databases that have been described in the database issue. So this is a definitive collection of maintained databases. There are something like 1,800. If you review them, actually, there are many connections into what's happening in ENA and its partners through the international exchange, INSDC. There are very few, less than 0.4% of the databases are not connected in some way to what's happening in INSDC, and something like 45% are directly connected. And so I show this really to say that once data, and this includes viral data and host COVID data, arrive in ENA, there is automatically, because of the openness and because of the interfaces that we provide, there is a spread of those data, an adding of value, further curation, further analysis, and this allows us to achieve a great deal, not least the COVID-19 data portal. So on to the next slide, please. So I'm going to switch to the SARS-CoV-2 data hubs component of the European COVID-19 data platform. So this is a toolbox that is there for people who are working with viral sequence data. So it allows people to organize their data, to run cloud analysis, and to share and to make publicly available the data and the analysis that they have. It's based on a system that pre-existed COVID, called the Compared Data Hub system, but we've extended and deployed very specific tools in it. So there are some data upload or enhanced data upload functions. Nadine will mention some of those. We have tools to validate against data standards, so a simple but I think effective data reporting standard emerged very quickly around viral sequencing data. We have a number of different workflows according to which sequencing platform is being used, according to which library method is being used, and then we have different environments for exploring and analyzing or looking at the outputs of those of the different workflows. And so one of the things we really wanted to prioritize early is making sure that that we could understand variation accurately across different viral isolates. Early on, there was very little sharing of raw sequence data. People were beginning to share some of the assembled sequences, but already in assembly, you make assumptions about how to handle the sequencing error. So really, to understand true variation, which is becoming ever more important in SARS-CoV-2, one needs a systematic approach to understand, to process the raw data. And so an early priority was to collect these raw data and to make these available for systematic analysis. And if you look at the bottom right, these are the kind of workflows that we imagined, and we and others are beginning to build these. So at this point, I hand over to Nadine.
Yes, thank you, Guy. If you go, yeah, thank you very much. So I'm going to talk about the data mobilization efforts as part of the European COVID-19 data platform that Guy introduced early on in the slides. So this involved at first to outreach to EMBL member states and different institutes and groups that were involved or coordinating SARS-CoV-2 sequencing within different countries. And the aim of this really was to identify barriers to sharing data and support in trying to overcome these barriers. And so initially this started off with things like providing help and guides for SARS-CoV-2 submissions in the form of documentation, but also in the form of providing direct points of access for users to contact members of the EBI. So the virus data flow email and the e-COVID-19 emails, both available from the COVID-19 data portal, and related to submission and data sharing support and also collaboration requests. So we started this outreach in around March of 2020. There were also things like support in setting up submission routes into the ENA and supporting data sharing in that aspect as well. So we've wanted to see at this point what's happened to from this, or what's come about from this data mobilization. So if you can hit next, please. Thank you. So one of the ways that we can see this is through the sheer numbers of the amount of data that has been shared and submitted, in particular, for example, to INSDC. So these two graphs that you see present the numbers of raw read data sets that have been submitted to INSDC in the top left, and then on the bottom right, you see the assembled sequence data that has been submitted to INSDC. What you can see from the raw reads is that there is a real increase from around May of 2020, and it's a very exponential like curve that you could see. And this has corresponded to around more than 225,000 raw read data sets that have been archived and deposited with INSDC at this point in January of 2021. So this is where a lot of our data mobilizations have been, efforts have been focused towards, but we also have seen more than 45,000 assembled sequences that have been submitted. And the graph at the bottom right is slightly out of date. It stops at November of 2020, but you can go to the COVID-19 portal and view current and up-to-date stats on this. And there's data mobilization efforts are also starting to focus on supporting assembled sequence submissions. So the numbers should increase hopefully soon in the coming months. So if you go, thank you very much. So looking at the geographical spread of this from the data mobilization efforts, it might be a little bit unclear, but I'll go through the map that you're seeing there. So the darker the color of orange that you see there, the greater number of submissions of raw read data sets to ENA or as part of the data mobilization efforts. So what you can see here is that there is a large center of European data that has been submitted and shared, but we will also have reached out to other non-European and EU member states as well. And in addition to that, we've seen, as part of data mobilization efforts, data from Brazil, Malaysia, and Thailand as well there. So if you can go to the next slide, please. Thank you. So the other aspect is that as we are part of INSDC, it means that we also have access to other data from our partners, such as NCBI, and this expands the global reach of our data mobilization efforts in this case. But what I want to point out here as well is that for the data mobilization efforts that's included here, around 3/4 to 4/5 of the data sets that have been submitted through data mobilization efforts have really made up the entire COVID-19 data sets that you're seeing, for example, in this map on the slide here. So if you can go to the next slide, please. Thank you. So some of the tools that have been developed as part of the efforts include the drag and drop uploader tool. So this was developed in April of 2020, and its aim, so submitters here can obtain a submission key, prepare a metadata spreadsheet along with data files, and then, as you can see on the screenshot, drag and drop them into the box, hit upload files, and then submit files, and then a user support bioinformatician at the ENA carries out the submission on behalf of the submitter. There is ongoing backend automation so as to make the steps for the user support bioinformatician automated and from start to finish. And we've seen 9 submissions from start to finish so far, and there's many more ongoing. And every month there are new submissions that we're seeing as well, new requests that we're seeing. There's a couple of examples that I've presented there, and the second one in particular shows that the tool scales relatively well with larger data sets as well. So this data set includes raw reads of around 400 data sets there within the project, entirely submitted through the data uploader. So for more information on that, you can contact the Virus Data Flow email and also obtain a submission key if you're interested to submit. Thank you. So some other tools include the GIS-aid submission conversion tool. So this is a tool that has been developed to support submitters in submitting their assembled sequence data to GIS-aid, but also in addition to ENA, and make this process relatively, or support the users in this process. So this will convert a GIS-aid submission spreadsheet into one that is compatible with ENA, in particular for ENA submission through WebIn-CLI. So this is a shout out to some of the other existing submission services that we have. So WebIn Interactive and the submissions portal for interactive submission, WebIn REST for programmatic submission, WebIn-CLI, which is a command line interface which validates data before submission into ENA. And we've also developed a tool that wraps around this WebIn-CLI tool that enables for bulk submission for the user, that starts from a spreadsheet and carries out and orchestrates that submission. So next slide, please. Thank you. So as you can see here, that I've talked about some of the tools. I've talked about the archival of data. Now I'm going to talk a little bit just on this slide about the presentation and discovery and retrieval of data as well. So one of the ways to access this data is through the COVID-19 data portal. So the link there will take you directly to the viral sequences section of the portal, and there are several options that I think Guy has mentioned earlier as well. So including the interactive downloading feature, retrieval from API using an API, and then the bulk downloads, which enables users to download the sequences and raw reads for everything that's presenting in the COVID-19 data portal at the moment. And just to reiterate, the portal is an access point for a wide range of data types, as Guy mentioned, for spanning across the different resources at EBI. So there's ways to share other types of data here as well, mentioned as shown in the screenshot on the top right, where you can go to submit data. Thank you. And so just to bring things back into the context and provide an example use case here. So what myself and Guy have been talking about is that first bit on the left-hand side where you see SARS-CoV-2 data hubs and COVID-19 data portal. But here, the sharing of data here really enables for, so at the bottom you can see data mobilization and access of data, enables for other analytical workflows to be run on these open and shared data, and then visualization and navigation as well. So here we've shown the Galaxy project and also the CRG beacon, which other users can plug it into as well. Yeah, so I think that concludes this side of
Thank you, Nadim. And with that, I give the word to Björn Grüning to talk about how we did COVID-19 analysis in Galaxy.
Yes, thanks a lot. Thanks a lot, Nadim and Guy. Yeah, coming back to this initial figure, so we have covered more or less 10 parts, so data storage, and also how we share data, how we share access, and how the entire open infrastructure has improved data access during the last year. We will now concentrate a little bit on the middle part, so data analysis. And I will talk a little bit what we have done in the Galaxy project. So the global Galaxy project in my next slides. Next slide, please. So on this website, the COVID-19 Galaxy project.org website, we try to collect more or less all workflows, all tools, all packages, all containers that are kind of relevant to analyze data sets. So everything that we have here is publicly available on GitHub. There are 6 different types of data. So we will more or less focus on genomics here, but you will see that that there are many different types, like cheminformatics or proteomics, which you can use to analyze, or Galaxy can be used to analyze those data sets on different types of data analyzers possible with Galaxy. We have teamed up with a bunch of other Galaxy systems, including the one from Galaxy Australia that you will hear in a few minutes. And our aim is, it's probably also the only way how we can actually reach all the scientists across the world, is that all these workflows are actually distributed across different Galaxy instances, across different infrastructures, but with the reproducibility in mind. So essentially what we are trying here, we try to develop best practice workflows together with clinicians, together with virologists, with scientists, but then push those workflows out to larger infrastructures, to public Galaxy servers across the world, so that everyone can actually participate on that. And ideally not only analyze their own data, but access actually the public data sets that ENA and others provide. Reproducibility is a key, and just to make sure, no, you can go, it's okay. Just to make one point clear, even if we are only talking about ENA here, Guy already mentioned there are many other databases like Pride that is responsible to share public proteomic data sets. And there are initiatives and there are also workflows that completely deal with proteomics. So a shout out here to the Galaxy proteomics team that actually analyzed or reanalyzed publicly available data sets on Pride. And we're actually able to find new things in public data. And this is actually what we would like to encourage, right? So people should share data. People should be able to access all different kinds of data, should put them together, like in these meta-proteomic analyzers, and maybe draw new conclusions. So this is more or less just next to sequencing, there are different communities that the Galaxy community is also covering. A lot what we have done in the last, since our last webinar, is actually about new tools and workflows. So obviously the field has moved on. There are new tools developed, bugs have fixed. So we put a lot of effort actually to update these tools constantly, so you can also get constantly the newest tools and the newest workflow iterations. We have also integrated completely new tools in the sense of we can now also from Galaxy push data back to ENA and public archives. We will cover that in, I think, next week. But we also have tools that actually can get data easily in Galaxy. For example, if you have your data in EGA, so the European Phenotype Archive, you can now get these data with your credentials into Galaxy and you can analyze them. The figure that I have put in here is actually a nice example of how interdisciplinary research should work. So this plot, and actually the tool behind this plot, was actually developed by a virologist here in Freiburg in our university hospital working on COVID data. So he is using the Galaxy workflows, but was kind of missing such a plot. So he developed this plot, we integrated it in Galaxy, and now everyone can access this new plotting tool dedicated for COVID research. And what you are seeing here are different strains across different strains from different time points. So every row is one of these strains, and you can more or less very easily see the variance between those different strains. Yeah, and there are a bunch of more tools, so we cannot list them all here, but we have iterated on all of them and all on our workflows. Please, next slide. And I just want to highlight two of these workflows. So in the beginning of the pandemic, the data that were published were to a large degree Illumina data sets, whole genome data sets, and so on. That has shifted a little bit in the sense that most of the data sets that we have freely accessible now, I would say, are Arctic data sets, but also the amount of nanopore sequencing technologies or nanopore sequencing data sets has increased. So this is one data set specifically for Arctic and ONT data set that we have developed, and that is also now freely available. Next slide, please. And Nadim and Guy already talked about easily construction of, or the consensus construction of genomes that then are deposited in GIS8 or hopefully in ENA or public archive. But this is the common needs. And I know, at least from Germany, that actually the government requires some kind of construction or some kind of consensus sequence that needs to be constructed. So what we created over the last is also those kind of consensus workflow. So you can, if you are finished with your variant calling, you can you can just use that workflow on top of your variant calling workflow. And the output will be a consensus sequence of the virus that you can then push to archives, or if you need to, you can also push that to GIS8. Thanks a lot for Wolfgang for creating those workflows. All of these workflows that you can access, and these are by far more than 20 different workflows now, are all deposited in Workflow Hub. But more importantly, if we use these workflows on public infrastructures and process them on publicly datasets like from ENA, you get actually the entire provenance stored in such a history. So you can actually look up how these datasets were processed, which tool versions you have used, which enables a very high degree on transparency and reproducibility. Maybe more interesting for researchers is even you can access those histories and just download the VCF files on your own. You can maybe just pull down the BAM files if that is the interesting piece that you want. Or what we also offer now is pre-computed VCF files for all pre-analyzed sequences. So if people upload data to ENA, we have a system in place where we automatically process those data sets nowadays and give you more or less the VCF files, the variant calling files, the analyzed virus variants as download. And you can then, take these 80,000, 100,000 of VCF files and process and do your research on them, and hopefully come to new conclusions that help us to fight this virus. Next slide, please. One thing that we have, that we needed to learn over the last 6 months, I would say, is that if you have access to 100,000, 200,000 data sets, even the download becomes cumbersome. So even with a university internet, it takes time, but even worse is when the download aborts or you need to restart downloads and so on. So it's not so easy to get actually these 200,000 data sets copied. So, and to avoid that every user tries to do that, what we are doing is we are mirroring more or less the COVID-19 data portal, at least the sequencing part and a few of the proteomic data sets, and put them into a Galaxy data library. And this data library is then shared with every user on this Galaxy instance, so you can access them directly and you can run your workflows on this data sets, which is more or less a mirrored copy of the data in the COVID-19 data portal / ENA. Next slide, please. I guess my main point, what I would like to point out, and the main difference between all these updates that we have and all these infrastructure that we also extended over the last months, and we are very happy to new partners that joined our distributed compute network, which Andrew will talk a little bit more about that. But my main point here is, I guess, when our last webinar was more or less about, hey, we have a nice workbench where you can analyze your data sets, I think we are shifting, and this is actually a good thing, we are shifting to, hey, we need to monitor. We need to monitor the outbreak. We need to constantly run our workflows on new data sets to actually, as soon as possible, find new mutations. This is technically not a huge shift, but monitoring becomes more and more important. And if you look at the last webinars, that's kind of what we expected, that as soon as a vaccine is there and as soon as there's more pressure on the virus, that it will evolve faster, and we need to monitor those variants. This is at least my slide until I will hand over to Andrew from Australia. I just want to make this point that the infrastructure that we develop here, and that also Guy mentioned, is actually infrastructure that we roll out across Europe. Right? Europe is big, but we are aiming even bigger, and this infrastructure is deployed in US, and we are working together with the US. This infrastructure is deployed in Australia, and we are all using the same technology stack here to improve the global fight against COVID-19. And with that, I would like to hand over to Andrew. And thanks, Andrew, especially because it's now 3 a.m. in Melbourne. So thanks for joining us.
Very happy to be here. Thanks, Björn. Next slide, thanks. And the next one. Yeah, so thanks, Björn. That's a really nice introduction. I'm going to talk specifically about Galaxy Australia. So Björn mentioned we have now a global infrastructure. It's very, very powerful, what we're able to do, given our agreement, if you like, at different levels of infrastructure globally. And now Galaxy is a very powerful platform. And if I can go to the next slide, and then I might actually go to the next one after that. Galaxy is extremely powerful in allowing people to have a consistent interface to a global set of tools and workflows. And at the start of the pandemic, way back, it seems like about 5 years ago, but it was actually only in February, really, in 2020. Of course, there was an enormous amount of effort that went into understanding COVID-19. Lots and lots of preprints. Wasn't a lot of public data sharing or public method sharing. And in fact, there was a lot of inconsistency in the analyses that were happening and the data that were being shared. So we realized that we already had a long-standing agreement in making a very standard-spaced analysis platform available to a wide number of researchers using what we call usegalaxy.star, which is a set of consistent Galaxy servers around the world. And if I go to the next slide, What it allowed us to do is to leverage what we'll call the basic unit of sharing and openness, if you like, in Galaxy, which is the workflow. And here is a workflow that's used for analysing COVID nucleotide data. We were able to very quickly set up an infrastructure, a global infrastructure, where researchers from different countries, including Australia and Europe and the US, where Galaxy Main is based, could share the same workflows with a guarantee that they would get the same outcome from running that workflow on the same data, and that was actually quite a big leap. This is an example of a nucleotide, a COVID nucleotide analysis workflow, and here's the set of workflows. Sorry, next slide. Thanks, Frederick. That we shared way back in February, and then a few more have been available. You can see the dates there on the right. Around the sorts of things that you would expect that we would be sharing, nucleotide analysis workflows on, read pre-processing assembly. Most common recent answers are variation analysis and whatnot. And if I go to the next slide, these are the outputs of those different workflows. So pre-processing, assembly, and you can see some examples of graphical examples of assembly of COVID sequence data there. Most recent common ancestor timing, variation analysis, and leading on to evolutionary analysis of the virus. So I just wanted to quickly talk about how quickly this happened, and it was in February 22nd, I think, was the date on some of those workflows. I actually got contacted on a Friday morning by Frederick and Björn and the folk from the US saying, we are doing this co-analysis of COVID across at least the US and EU at the moment. Can Australia be part of this? So I said yes, and 2 days later we had a complete replication of all the analyses of the fairly small number of COVID complete genome sequences that are available at that stage. And that is really amazing, right? So within 2 days we had gone from this happening once to this happening multiple times, reproducibly, transparently, across 3 continents. That was really amazing. Next slide, please. Next slide. Now this is the outcome of that, a paper which you can in PLOS pathogens, sorry, slide before, which you can look up at that URL down the bottom there. Again, it was for me, it was quite astonishing that we were able to demonstrate reproducible, quite sophisticated science underpinning COVID research in 3 continents in the space of days. And as we do, being academics, we published it. Okay, next slide. So the workflow is the unit of sharing in Galaxy, but Galaxy also allows us to share compute resources seamlessly between both within countries and between countries using this technology called Pulsar. And this is really, really important because COVID is such a powerful political term, and it still is, and it certainly was back then. It was actually fairly easy to loosen up, if you like, or free up some quite significant compute resources on the back of COVID research. Now, freeing up those compute resources and making them useful are 2 different things. But this technology, this Pulsar technology, which Galaxy leverages, enabled us to essentially add in as much more compute resource, scalable compute resource, underneath that sort of level of workflow transparency as we wanted. And we were able to, for instance, tap one of our supercomputing centres in Australia, which is called the Pulsar Supercomputing Centre, and they gave us quite a large allocation, which we were able to make available again within days to all of the researchers who are coming through the Galaxy portal, the Galaxy Australia portal. Not just that, but we were able to actually use some of those resources through the Pulsar standard to make the compute in Australia available to use Galaxy.eu. So that was the power of agreeing on that standard of resource sharing, not just workflow sharing, is that we could free up resources from Australia, make them available to a large number of researchers in Australia almost immediately, and also make them part of the global pool of compute resources, which was necessary to make the research as scalable as it needed to be, particularly as Björn was talking about when we're talking about cheminformatics and things like that. Next slide, please. Okay, here's an example of the Pulsar resources that we made available in Perth, which is a city in Western Australia. And we were able to just attach worker nodes as we needed to, and they all have the same consistent interface which Galaxy makes available. Pawsey made some resources available in their supercomputing centre, and Galaxy Australia was able to send COVID-related jobs to Pawsey that very same day because we were able to leverage the standard-based resource sharing. Next slide, please. Okay, so just wrapping up then, here are some statistics around what we've been doing. These are a little bit old, and of course, there's been a lot more happening since then. We've uploaded about 15,000 sequences to the European Nucleotide Archive, and that's part of our contribution to the global pool, I guess, of COVID sequences that Nadim and Guy talked about earlier. We've got lots and lots of COVID-related jobs being processed, of course, through our supercomputing centres around the country, and it's specifically in Victoria, which is a state within Australia, in which I'm talking from Melbourne, which is the capital of that state. We've processed 2,000 COVID samples through the Arctic workflows, which are again exactly consistent with the same Arctic analysis that's been happening throughout Europe. I'll leave it with that and hand over to Frederick. Thanks very much.
Thanks, Andrew. And I will try to wrap things up by placing all what you've seen today into an integrated ecosystem that we've built to really make this as seamless as possible. For this, I want to introduce, in case you don't know, the European Open Science Cloud. This is one of the major drivers for open data and open science in Europe. And its aim is to create a virtual environment where we can seamlessly integrate different services based on the standards that we have, and do this across scientific disciplines and across national borders. And there are similar projects or similar programs in the US. Anvil is such a project, for example, and a lot of other countries have similar aims to really drive open science forward. And then COVID is often an important proof of concept or pilot in these things. So if we deconstruct this a little bit, how does this map to what we've done to Galaxy? So in terms of the standardization, we are building on an infrastructure that's there for already a long time. Within EOSC Live, which is the project in Europe to build the European Open Science Cloud for life sciences, we have focused on a number of technologies which are also underlying Galaxy. Conda and BioConda for tool packaging, Docker or Singularity containers, and a lot of the registries that Elixir is maintaining are crucial for that. What I want to highlight here is all the different standards that we're using to make this interoperable. We are using the Open Container Initiative around container standardization. For workflows, we are focusing on common workflow language to align the metadata standard, and we package the workflows through research object crate. Tools are described by EDAM ontology, so we can leverage this common language across the different registries. And for example, bioschemas, which is an extension of schema.org used by all the major search engines, allows us to find all this data easily through the ways that we are used to. And we do this in collaboration with entities such as the Global Alliance for Genomics and Health that are focusing on developing new standards and further implementing those. Similarly, within Elixir, we have the tools ecosystem. Again, an ecosystem that existed before this pandemic hit, where we have all the pieces that we need for containers, tools, the necessary benchmarking, and workflow management systems such as Galaxy to make this all happen. One thing that we added in last year was the workflow hub. So I just want to highlight this here. This is a workflow registry. This is part of the EOSC Live project. And because of COVID, we fast-tracked this actually to be able to host the COVID workflows and easily share them. The idea is that workflows remain in the host repositories. We can organize this based on the workflow management system or, for example, the topic. And we can associate all relevant information. And we're developing this, and we've been doing this for the last 8 months, more or less, in partnership with the workflow management systems, Galaxy and others, to make it as easy and integrated as possible that you can submit workflows from Galaxy to the workflow hub without having to jump through hoops. And we can do this for Galaxy and other workflow management systems because we are using these standards. So I think we can check based on standardization and the virtual environment part of this. So if we look at the cross-scientific disciplines and borders, already was mentioned, for the COVID-19 data portal, there are a lot of different data types. Also within Galaxy, we have these workflows in different things, not just genomics, but also proteomics or the cheminformatics part. And in some of the webinars that will follow, we will go into more detail on those. And Galaxy as a whole is even broader. Also climate, for example, or ecology are covered by the training materials, which is a fantastic resource that's available, where you can also find the COVID-specific workflows with a lot of detail on what they do and how you can utilize those. As was already shown, we have this use Galaxy.star collaboration, which allows us to go cross-border. And this is only one way that you can use these workflows in Galaxy. Our Italian Elixir colleagues have set up a service, Laniakea, where you can spin up on-demand Galaxy instances, and also an instance with the COVID workflows is available on their platform. We have Docker containers and Singularity containers available that you can deploy on your own infrastructure from your laptop to your local computing environment. And you can also build it from the source if you really want to control all the aspects. So we have many options to deploy Galaxy and utilize these workflows. And as mentioned already, we have this collaboration across the globe with aligned use Galaxy services. And if we go in a bit more detail, in Europe, we have several national ones that have been set up in the past year to specifically serve the national communities. So this community is really expanding a lot now. And then, as was also already highlighted, the Pulsar network really makes it very easy to go global and utilize resources from different countries. This also allows countries to contribute with, for example, compute resources while they don't have the manpower or resources to set up their own Galaxy instance. So this is really allowing collaboration. And as Andrew said, in the COVID pandemic, it was relatively easy to get people to contribute and donate these resources. So the cross-scientific disciplines and borders aspect we also have covered. Then the probably most difficult one is the seamless integration of all these services. There's a number of aspects around this, from storage and management of data to the analysis, and ultimately we want to reuse all of this data. There were already excellent examples in the previous speakers' talks. Just some highlights here. So with the COVID-19 data portal, we have this way of mobilizing the data, getting access to the data, and working with the national SARS-CoV-2 data hubs to gather this all in one place. We've made links with Galaxy to be able to analyze this data seamlessly. And for example, the integration of the CRG viral beacon in Barcelona allows us to reuse these data and visualize it in a different way and bring it all together. One thing I want to highlight here is this connection that we've made between the COVID-19 data portal and Galaxy. So as Björn also mentioned, so we can automatically mirror all the data and align what is available in the portal with what is available in Galaxy. We've been collaborating with EBI to make sure that these data sets match. And we've also implemented tools that allow to do submission of viral data with some pre-processing if necessary to ENA and the COVID-19 data portal from Galaxy. This will be explained in more detail next week in the webinar that we will be hosting. And then as a final slide, I think we can really see the infrastructure that we've shown today as an exemplar implementation of the European Open Science Cloud and all of these similar projects across the globe. Because we did this not just in Europe, but in a global context. And the only way we could do this in the time frame, as Andrew explained, of a matter of days rolling this out globally is because we're building on an existing open and standard-based infrastructure. And it's been really amazing, this collaboration. It's unfortunately, there's a pandemic and that requires this, but it has been fantastic to work together and really make this happen in a collaborative way. With that, I want to acknowledge all the funding sources that we have. There's a lot of projects that have been dedicated to COVID research and this specifically, and I want to open up the floor for questions.